Hello. We can't see me and I don't know why. Hmm. Here I am. Can everybody hear me okay? Let me turn on my light. Hmm. Dun, 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 dun. Total amateur hour over here. Here we go. Okay. How's my audio? Is my audio good? Steven says good. And if it wasn't good, Steven would have to fix it. That's how that goes. Hello, everyone. I'm back. I was in Costa Rica last week. For those of you who don't know, I was on vacation. It was a little bit rough. We still managed to have a good time, but I was sick for about half the trip. So I had some sort of stomach ailment, lots of throwing up, running fevers, not good. My pants are fitting really loose, so I guess that's good. But yeah, difficult times. But Costa Rica is a really beautiful place. I highly recommend it. It's a great country. We went to a place called Manuel Antonio, which is a national park that's pretty famous there, and we saw lots of animals. We saw sloths and two kinds of monkeys, two kinds of sloths, two-toed and three-toed. We saw like the little Jesus lizard run across the water, lots of iguanas by the ocean. So it was pretty cool. All the animals we saw, and the beaches there are really, really nice. So we had a good time. I just unfortunately was sick for half of my vacation, which is not very fun. So, but I'm glad to be back and excited about tonight's live stream topic. So, yeah. But before I start that, I have an announcement. I'll be at the Keystone Clash speaking, which you knew, but now I am giving 2 talks and 2 panel chats. So my talks, one of them will be CO2 for beginners, and one of them will be on glowfish, glowfish genetics. So if you have pictures of glowfish, if you've ever kept them, send them to me, please. And then I'm doing 2 panel chats. So one panel chat will be with Dan of Dan's Fish and Bentley Pasco, and we're going to talk about rainbows. And then the other one will be with Ted Coletti, who is one of my personal heroes, and 2 other people I don't know, Sue and someone named Peter. They are club members with Ted, and they are pond people, and we're going to be talking about tub ponds, which is something I'm also super passionate about. So that should be a lot of fun. And actually, next week, Stephen and I and Myrtle on What Your Plants are going to talk about ponds. We're going to talk about tub ponds because I do them every year, and I'm very excited about them. And it's the happiest thing I do all year are my tub ponds. So we're going to talk about that. So that'll be Monday, 5 p.m. Eastern time. So, but tonight's topic is very exciting. We are talking about phylogenetic trees. So phylogenetic trees are they are diagrams that show evolutionary relationships. And you've probably seen them before. You've maybe seen them at fish talks, or maybe you've looked up papers on your favorite fish before, and you see these diagrams and you're like, well, what are these things? How do I read them? So we're going to talk about a little bit about how to read them and interpret them, and then we'll talk about how they're made, because there are different methods for making them, and it's kind of interesting. So let me just get my PowerPoint started. from beginning. All right. Hang on, let me share my screen. Share screen. Share screen. And oh, why is this not window? Share. Okay. So let's get this started. All right, so phylogenetic trees. So this is just an example of a phylogenetic tree. This one is of animals. I just picked this as an example because we all love animals. And so this is something you might see pretty often. So a phylogenetic tree is a diagram that shows evolutionary relationships. And this is made by comparing different characters. So what are the characters they're comparing? Well, you could, if you have a phylogeny of dinosaurs, maybe you're looking at teeth. Maybe you're measuring teeth. Or maybe you're looking at number of vertebrae. Or if you're looking at modern organisms that are still living, maybe you're looking at DNA sequences. And so things that are grouped together. So for instance, here we have hemichordata and echinodermata. They are grouped together. And so they are more closely related than things that are chordates. So chordata. So, and these three right here are more closely related than this clump right here. So gastropods and bivalves and annelids. So, oh, come back. We didn't mean to do that yet. So in a true phylogenetic tree, the length of the line is proportional to the number of changes that happen, the number of characters that have changed. So the number of base pairs of DNA that have changed, or like the number of vertebrae that have changed, or whatever change you're measuring. So you can see that like this line on gastropod. So there's been a lot of changes that have undergone in gastropoda since it diverged from bivalves. So they share a common ancestor. The common ancestor would be at this node right here, and they're related. So you wouldn't say that bivalves evolved from gastropods or vice versa. You would say that gastropods and bivalves share a common ancestor. So that's just like you didn't evolve from your cousin. You and your cousin share a common ancestor. Your common ancestor would be your grandma and grandpa. So that's what this is. So, and gastropods are like snails. We have to talk about snails because we have very many snail lovers in the chat. And bivalves would be clams. So there's been a lot of changes since that divergence happened. So this is just one example of a phylogenetic tree. This is focusing on metazoans, which are true animals. So parazoans are sponges. Just FYI, they are not true animals because they don't have true tissues. So way back in episode 1 of Science is Fundamental, we talked about tissue differentiation, and we talked about the germ layers that would form. So sponges don't have that. They're just an unorganized mass of cells. So these are metazoans right here. And all metazoans share a common ancestor. And they actually share a common ancestor with sponges who have gone on to evolve in their own way. And so this tree right here is what's called a rooted tree. And I know it's rooted because it has an outgroup. So an outgroup is something that you choose when you make your tree that you know is very, very distantly related to everything. So for instance, if I was doing a phylogenetic tree of my family, with my cousins and my second cousins and all that, like I could use like Stephen as my outgroup because I don't think Stephen and I are very closely related. At least I don't think so. So that would be the outgroup. But in this tree, the outgroup are excavata, which is like Giardia. If you've ever heard of that, that's a single cell parasite, Giardia. So, and also my good friend Cryptosporidium is a member of excavata. And Cryptosporidium is a tropical disease I had once and I was hospitalized for it. So, it's a good friend of mine. But so what else can I tell you about phylogenetic trees when you read them? So you can come back, stop that. You can rotate any of these symmetrically about the node and it means the same thing. So let's say I had this little group right here. So a group is known as a clade. So I had this little clade right here, the hemichordates and echinodermata. So echinodermata would be like starfish and like sea urchins and sand dollars, and hemichordates, those are tunicates. Okay, so you could take this clade and you could switch these two and it would mean exactly the same thing because you can rotate anything about a node. So if you were thinking three-dimensionally, you could just pick that up and rotate it and it would mean the exact same thing. And you can do that with any node and it'll be symmetrical. It'll mean the same thing. It's just how you want to present the information, but it's identical. Other things about this tree. So trees are built using shared derived characteristics. So
Sorry to interrupt this program, but people in chat are complaining that they can't see shit. Make it bigger.
oh my God, I'm sorry. I don't know if I can make it bigger. I'm sorry. Yeah. I don't know that I can make it bigger. I'm really sorry. Let's see if I can escape.
There's a zoom button at the bottom of PowerPoint on the bottom right. You can move that slider
Yeah. Hang on.
and zoom in.
Oh yeah. How's that?
All right, perfect. Okay,
All right.
voice of
Sorry
God.
about that, guys. I mean, unfortunately, there's just a lot of information on these trees, and they're always like vertical, and they're horizontal. So I'm sorry, but thank you, Stephen, for being a good admin and keeping it on that. So, anyways, you can rotate any of these about a single node, and they're symmetrical. So it's just how they chose to present it. They could have just as easily switched hemichordates and echinoderms and it would have been the same thing. They could have switched this whole group with this whole group and it would have meant the same thing. So trees are built on shared derived characteristics. So what does that mean? It means that everything is in a hemichordate has accumulated the same evolutionary changes and they have that in common. So it's divergent evolution. So you have a common ancestor. The common ancestor would have been way, way back in evolutionary time. And some of them took one path and some of them took another path. And all the ones that took that path, we would group together. So all the hemichordates are related and all the echinoderms are related. So there's other kinds of evolutionary changes that would happen, but they are not informative to making trees. So I'm going to make this smaller so you can see this one because this one actually fits on a slide. There we go. So convergent evolution versus divergent evolution. So we want to use divergent evolution for making trees. So let's say that we are looking at a piece of DNA and we have an adenine and one of them takes a path. Well, this would be amino acids because there's no bases that begin with S. So let's say this is an alanine and it diverges to become can't remember what amino acid begins with an S. Well, whatever. It makes that change. And then another path, it makes that change. But convergent evolution is where you have two things that are not related that converge on the same path to become the same thing. So this is not an evolutionary relationship. These just evolved this way because they had the same sort of pressures, same sort of selective pressures to cause it. And there's lots and lots of examples of convergent evolution, and you know about a lot of them. So for instance, echolocation has evolved multiple times in animals. So bats have it and whales have it, and they evolved it independently. Um, so you can't use echolocation as a character to make a phylogenetic tree because it is not a shared derived character. It is convergent evolution. And there's lots of examples of it. So for instance, there's lots of Australian marsupials that look a lot like placental mammals, but their common ancestor didn't look that way. This is an example of convergent evolution. So for instance, there's a regular mole, the mole that would, dig up your garden. And apparently there's a marsupial mole too. I mean, I've never seen one. I don't live in Australia, but maybe Mark Searleson can tell us about these. But apparently there's something that looks like an anteater that's a marsupial. There's a marsupial mouse. And I've seen pictures of those and they're very, very cute. But there's something that looks like a lemur. There's something that looks like a flying squirrel called a flying phalanger. I don't know what that is. I don't know if that's a sugar glider. I don't know. I am not an expert on all animals. Cats. So the Tasmanian tiger is extinct, but it looks a lot like an ocelot. And of course, the Tasmanian wolf looks like a regular wolf, and that's all convergent evolution. They evolved independently, so it is not a shared relationship. So you can't use those to make a tree. You can only use divergence to make a tree. And then this happens in plants a lot too. So on the left, this is an agave, and agaves are native to North America. And then this is an aloe plant, which looks a lot like an agave. And there are lots of species of aloes, and some of them they look exactly like agaves, but these are from the Mediterranean. So they evolved looking like that independently. And Eric Wyrock, I see in the chat right there. Yes, dolphins and ichthyosaurs, they do look very similar, and that is indeed convergent evolution because the common ancestor of both of those look nothing like that. So they evolved that body shape independently, and there's just tons and tons of examples of convergence. And it's just because similar habitats select for similar organisms, because this body plan, for instance, right here, with the aloe vera, this is really good for deserts and dry, hot climates. And the Mexico and the Mediterranean have really similar climates, really hot and dry. And so that's why you would see plants that look really similar in both of them. But you cannot use this to make a phylogenetic tree. You can only use divergence. And then there's something called parallel evolution, where you have two paths and they do the same thing. And you can't use parallel evolution to make a tree either, because you can't tell things apart that way. So you're always looking for shared derived characteristics. So differences that come from a common ancestor. So that's what a phylogenetic tree is. Well, how do you make one? So there are people who devote their careers to this. My very first job in science, I worked in a lab at Iowa State. I was 19 years old. I didn't have a job outside of science between the ages of about 19 and 34. And then I had to take a break, and I was a barista for a minute there because I was so sick of lab work. But my first science job, I was 19. I worked in a lab, and it was an evolution lab, and they worked on the evolution of cotton plants and things that are related to cotton plants. What we would do is we would sequence different segments of DNA, and Um, you would put this in a computer program, and this was the 90s, so these would take a long time to run, and software would make the phylogenetic trees for you, and it's based on like Bayesian mathematics and very complicated algorithms that I don't really understand because I'm not this kind of scientist. So, but it's pretty complicated how it's done. But the to do a really good phylogenetic tree, you need to look at lots and lots of characters. The more, the better. So if you're looking at fossils, you may not have very many informative characters that you can look at. So like, you can look at things like teeth. You can look at bones. There's tons of little measurements they make in the bones and in the vertebra. And, those scientists they're really amazing how they do that. And that's not anything I ever did. But if you're looking at modern organisms, really the best way to make a phylogenetic tree is to use DNA. And that's because with DNA, you have thousands and thousands and thousands and thousands of different characters you can look at. And you can choose different DNAs based on how long they've been diverged for, because different genes evolve at different rates. So if you're looking at a really ancient evolution, so like tree of life stuff, so we've talked about like our eukaryotes and prokaryotes and archaebacteria before, you would want to look at DNAs that evolve very, very slowly. But if you're looking at animals or plants or something that they've separated pretty recently in history, so for instance, African cichlids, they're evolving very rapidly, you would want to look at pieces of DNA that are changing all the time. So you would get more informative characters. And there's kind of a real art and a science as to which genes you're sequencing and which segments you're choosing. Um, so the classic one that's looked at is this gene called cytochrome C. So cytochrome C is important in respiration. It's in plants, it's in animals, it's in lots and lots of things, and it's absolutely crucial for life. It's involved in respiration. So anything that uses oxygen is going to have cytochrome C. And so since it's so incredibly important, it's going to evolve very, very slowly. And that's because most changes in cytochrome C are lethal for the organism. And so you're not going to see much. And so that's really useful for looking at things like the split between well, the switch between, like, if I were looking at the difference between chordates and this, I might look at something like cytochrome C because I'm looking at really, really ancient separations. But if I'm looking at African cichlids, so there are African cichlids that evolved only 10,000 years or less ago, which is just a blink of the eye in evolutionary time. This is no time at all. You would want to look at a segment of DNA that doesn't even code for a gene. It's just some random interspersing piece of DNA, and that's going to evolve really, really rapidly because there's no selection on it. So, and something you may have noticed if you're a fan of scientific names is they change a lot, and I know that's really annoying. I know that I was watching a live stream with Kenny and Danny, and they were complaining how they used to know all the names of the cichlids and they don't anymore because they've all changed. And the reason they've changed is because, as more genes are sequenced and you have more information to make phylogenetic trees, you may figure out that two things aren't actually a species, that they're not actually related. And so they have to change the name. So, yeah, it's annoying. I know when things change names, but the name is a reflection of the scientific history, the evolutionary history. And so that's why that has to happen. So thought I would show you guys one more tree. So I get a lot of questions about this. This is a very simplified tree, and I think you guys can all see it. I'll make it a little bigger though. So this is maybe a tiny bit smaller. This is a simplified tree of rainbow fish and their relatives. And you've probably heard before, if you know anything about rainbow fish, that pseudomagills are not true rainbow fish. And the reason for that is, if you look at the phylogeny of them, so Melanitania and Glossolepis and Chilatharina, those are very closely related. And so they would all be in the same family, all of them. And their next closest relatives are actually Eriatharina, which are the threadfin rainbows, or the threadfin, rainbow fish. So, and we don't usually even call those rainbows. And then Rhamnocentris are more distantly related from those. And then something called Cairnsicthus, which you don't really see in the hobby that often. That would be even less related than that. And all of these make up a group called the Melanitinidae. Like, it's hard for me to pronounce, but this is a single family right here, the Melanitinidae rainbows. And those were in Australia and New Guinea. So, which is where rainbows fish originate from. But up here we have our friends, the pseudomagills, and they are quite diverged from Melanitania type rainbows. And so these are generally called a different family, the pseudomagill family. So Pseudomagillidae. And these are closely related to Celebes rainbow fish. So that would be part of that family too. And the reason that they're not all called just lumped in rainbow fish is keeping them is actually really different. They kind of, they just, they have really different bodies. I mean, a pseudomagill and a rainbow fish, they look really different. And I don't think it's a good idea to keep them together either, because they, the bigger rainbows will really outcompete them for food, I think. So, and then on this tree, the outgroup would be the Madagascar rainbow fish, because it's the least related of all of them. So one more thing I want to tell you, the last thing I want to tell you about phylogenetic trees, even though the length of this line, it's showing a proportion of changes, what it doesn't tell you is time. So it doesn't tell you like this is 100 million years or this is 10,000 years. And a tree can't do that. You have to calibrate your what are called molecular clocks in other ways. So every single molecular clock beats at a different rate. So something like cytochrome C that I told you about, that gene that evolves really, really slowly, that molecular clock beats very, very slow because it accumulates changes at a very, very slow rate. Whereas other segments of DNA, they beat really quickly. So, but you can't you can't know like one change equals 100,000 years. Like you can't know that just from the DNA. You have to calibrate the clock in some way. And there are different ways to do that. So you can look at the fossil record. And you can look at different layers in which the fossils occur. And you could say, okay, we know this layer is this old and the fossils are there, and this layer is this old and the fossils are here. And so that's how we know how old it is. Or in this case, you can look at geographically. So Madagascar rainbows are in Madagascar. And this kind of rainbow, these are in Australia and New Guinea. Well, Madagascar used to be in a single landmass with those, and they know how long that broke off. And so they can calibrate the clock that way. And another way they can calibrate molecular clocks are using radioactive decay rates. So they know like different layers are a certain age because of the decay of different isotopes. So if you've ever heard of carbon dating of something, that's a way to calibrate a molecular clock too. Yeah, Corework says something Gondwana. Yeah, that landmass was called Gondwana land. So, and when all the continents were together, that was Pangea, and we know when that happened too. And you can calibrate molecular clocks that way too. So, yeah, or phylogenetic trees don't tell you time. You need to calibrate that with something else, but they can show you the amount of change that's happened proportionately. So, I think that's pretty much all I have to say about phylogenetic trees. I mean, this wasn't exactly my area of expertise. I did go to a million talks on these because I worked in a department that had a lot of evolutionary biologists. And so they would show a lot of trees. So I did have to know something about how they were made and how to interpret them. But, this wasn't exactly what I did. So, all right. Well, let's start looking at questions. So, and I'm sorry you guys couldn't see that earlier. My bad. Let me scroll up. All right. Fat Kitty is back. So Fat Kitty did not go to Costa Rica with me because Fat Kitty stayed home here, and one of my former students actually takes care of the cats, and she spoils them, and she brings them catnip and gets them super high. And they love getting high. So they were well cared for. All right. Audio looking good. Scrolling. Okay. All right. Alec asks, was I afflicted with Giardia down in Costa Rica? No, I don't know what I had. I probably had some sort of enterobacteria. It probably did not have norovirus, or I don't know how Jake could have escaped that because norovirus is so transmissible. But Jake and I ate exactly the same foods. Like the night before I got sick, we both had the mahi mahi dinner. He got his blackened, I got mine grilled. We had the exact same thing for lunch. We had the same thing for breakfast. I mean, it was ridiculous. We kept ordering the same things. He was completely fine. So I don't know what was wrong with me, but I was running a fever and I was not very happy. So it was not good. But last time I went to Costa Rica, I was fine the entire time I was there. And then when I got back, I got cryptosporidium, which is not a very fun disease. And I was hospitalized for over a week. And I lost like 11 pounds in 2 weeks. So, but don't worry, I gained it back. It was not good. Oh, that's right, Jenna. The S amino acid is serine. I had all of those memorized at one point. There's 20 amino acids, and like, I don't know, some things just don't stick in your brain. And I'm not a protein biochemist. And Eric asked about convergent evolution. There's so many cool examples of it. So, for instance, flight has convergently evolved many, many times. So, in insects and in bats and in birds, that's all evolved separately. Another cool one, eyes. That's convergent evolution. So, squid eyes and animal eyes, or I mean other mammal eyes, those have evolved independently so many times. They evolved independently in insects, independently in mollusks, independently in chordates like us. So pretty cool. But that's all convergence. It's all convergence. Interesting enough, though, the genes are the same. It's using the same pathways, but evolve them independently. Maybe we'll talk about that sometime. It's pretty cool. Fish Dreams is coming in hot. I love it. Love it. So Mickham says, so those implanted Monty Python killer rapids killed the Tasmanian tigers. Yeah, those went extinct in like what, the 20s or 30s maybe? I mean, Mark will have to tell us, like, are there like reported sightings of them? Like, like we would say we saw an alien or something, like, oh, I saw a Tasmanian tiger. I really did. So, DNA, RNA trees. You can make phylogenetic trees out of some RNAs. So actually, ribosomal RNA is ribosomes. So they're very important in making proteins, and they don't change much over evolutionary time because they are so important. They're very, very conserved. Usually, a change for a ribosome is going to be bad. So that's why they don't change much. So yeah, you can use that. You could also use tRNAs, but you can't use messenger RNAs because those are always different depending on the protein. But yeah, you can use RNA to make trees too. Usually you would sequence the DNA that coded for that RNA, because DNA is much more stable than RNA. It's much more easy to isolate. Sequencing RNA is not great. Sequencing DNA is pretty easy. I mean, I've done it. How hard can it be? So, Mickham says, I guess killifish ought to be interesting to analyze. Many separate collecting points locations for quite similar fish. Yeah, I'm sure. I don't know a ton about killifish, but there's so many varieties of them that I do think it's probably pretty cool. I don't know who the killifish experts are. I know who the rainbow experts are. That tree that I showed you is a condensed version by a guy named Peter Unmack. And he is in Australia, and he is the guy who does all of the phylogenetic trees of rainbows. So when he says that Melanitania are true rainbow fish and pseudomagills are not rainbow fish, that's the person whose work I'm citing. He is a stickler about that. So Mickham also says perch and other fish may change body shape depending on the predatory pressure in their habitat, perhaps only in a few generations. So is that a true genetic change, or are they just is that a developmental change? So if it's just a developmental change, that is not hereditary. It just means they have some plasticity. So for instance, human beings have gotten taller over time. That's not because our genes are taller. Like, it's not that we've selected necessarily for tall people. It's that we have better prenatal care and we eat more protein in our diets. And so we can get taller. So I don't know which that is. But they may be rapidly evolving. So cichlids are like the famous, there are lots of evolutionary biologists who study cichlids because they're evolving so rapidly. Some of them diverged only 10,000 years ago, and I cannot tell you how short of a time that is in evolution. It's nothing. It's just nothing. I mean, the Earth is 4.6 billion years old. Animals have been around or life has been on Earth for close to 4 billion. So 10,000 years is just nothing. It's not even a blink of an eye. So cichlids are really cool that way. And that's because there's so many niches, micro niches for the cichlids to fill. And there's a lot of sexual selection going on, which causes things to rapidly evolve. And I guess all the cichlid keepers in chat can tell you more about that than I do, but they have all kinds of really cool mating strategies and color changing to attract mates. And all those things cause a lot of quick adaptive radiation and speciation. So Corwick says they're like flow charts, but you can't make any decisions. I mean, yeah, kind of. They're kind of like flow charts. They don't tell you the time, but they do tell you the divergence that's happened. And so those lengths of the branches, they do mean something. Big Shrimpin says, how many neon tetras can I put in a 10-gallon? I mean, I'll tell you right now, less than 4 million. That's the definitive cutoff. It's something less than 4 million. So Rico says, hey, did you know Daniel Velez turns 25 tomorrow? Well, he's just a kid. He'll be old enough to rent a car. And I've heard someone else is maybe turning 25 tomorrow. I don't know who that could be, Rico. So Stevens is 40 neon tetras, then another 20 after you use 35 of the original ones. They are not robust. They're not robust fish. Geek Boy can't wait till he can have fresh mahi-mahi in April. Damn it, I love mahi-mahi, but I got so sick. Maybe I need a break from eating it. I managed to eat a lot of seafood on that trip because the seafood in Indiana is not great. It's very expensive and it's not very good. So whenever I go to the coast, like I only eat seafood. And it was really good there, but why did I get sick? Was it from the mahi-mahi or was it from a vegetable? Who can say? I mean, I did eat a lot of fruits and vegetables there. Nancy says my vacation diet plan sounds awful. You know what? It was not great. 0 stars. Do not recommend. Daniel says, wait, you mean I'm only 2 years older than you, Rico Stan? He's just a young kid. So Coraworks says, Steve, just use glow Daniels. They're a bit bigger, but you can put more of them in a smaller space before they start dying. And if you get them, please send me pictures of your glow Daniels because I'm giving a talk on them at the Keystone Clash. And I need your pictures. So send them my way. So Mickham says, I know one apisto expert not caring for shape or coloration when doing research on species. Color, morphs, and body shape is not trustworthy factors, according to him. That could be. So, for instance, there's a pretty famous crocodile paleontologist at the University of Iowa. His name was Chris Brochu. He gave really great talks. And he said that teeth are not a good evolutionary characteristic for crocs because their teeth all look the same and they just lose them and replace them all the time. And so they're not informative. Whereas other things like they would just, finding one tooth would have so many characteristics they could use. So it kind of all depends on what you're looking at, what characters are informative or not. And that's something evolutionary biologists that's kind of, part of the art to their science is knowing what to pick. Thank you. I'm sorry I got sick too. Thank you. So Coreworks says carcinization is a form of convergent evolution. I do not know what carcinization is. So Fish Dream says, what do you think of human health genetic testing on the market now? Well, I mean, I'm of multiple thoughts on it. I'll tell you right now, I have not done any 23andMe sequencing on myself, and I will not do it. And the reason for that is, these companies should be paying me for my data. I own that sequence. They don't. And I don't want it ever used against me. I own it and other people feel otherwise. Lots of people I know have gotten their 23andMe done. I think it's really valuable for treating genetic diseases. I think it, helps parents make good decisions on, if they should have kids or not. I think it's great for treating cancers. If you have cancers in your family, I think it's a great idea to get that, like the BRCA1, BRCA2 locus sequence, which is the breast cancer gene. I think it's a great, it saves lives getting that done. I think a lot of the information on 23 is kind of bullshit. 23andMe is really bullshit because it'll say, you'll get information back like it says, oh, there's an 80% chance I'm a night owl according to this. Well, I mean, it just means that you have loci that are associated with certain things. A lot of that is really kind of bullshitty pseudoscience. I mean, I would take a lot of that information with a grain of salt. So I guess I'm ambivalent about it, I would say, but I have not gotten mine done and I probably won't get it done unless I have a very good reason. Like if I had a cancer running in my family, or if I had cancer, I mean, God forbid, then they really need to know your sequence of your genome so they can treat you. Because cancer medicine now is all very, very individualized. It's personalized medicine. So maybe I'll change my mind later, you know. Oh, I miss you too, Foxanne. I missed you too, but as you can see, I did not tan. So funny story. We were on a bus on the way back from the beach, and there's this kind of like slightly nutty Canadian woman, and she looks at Jake and I, and she says, "Oh, are you English?" I'm like, no. I mean, and she said, "Oh, well, I just thought you two were so pale." It's like, no, we're just really white and we sunburn easily. Like, we have to wear sunscreen and hats at the beach. Like, I cover up I wear like a rash guard that goes down to my wrist so I don't sunburn. I always wear a little hat in the tropics. I wear sunscreen every day. That's because I burn really, really easy. And so does Jake. So we're just super white. We're not English. So CoreWorks says guppies have environmental triggers for their genetic coloration in subsequent generations based on predation and light levels and hiding spots. So those are not true evolutionary changes. That's just phenotypic plasticity. So that just means that they can grow in different ways based on their environment. Unless you are selecting for different fish, like let's say you're selecting for all the blue fish or something, or all the big fish or something, unless there's some selective pressure happening, it's not a true evolutionary change. That's just phenotypic plasticity, which means that different organisms can grow in different ways based on their environment. For instance, if I was growing up in the Middle Ages, I would not be 5'10". I would be 4'10", because I would not have had good prenatal care, and I would not have had grown up with the high protein diet I have. So it says Dr. Wilson J.E.M. Costa in Brazil is one of the huge Kili experts. Okay. Well, I will maybe look him up. I am not a Kili fish person. I'm a rainbow fish person. I think Kili fish are really cool, but you can't keep everything. And I only have 3 tanks. So the only Kili fish I've ever had are Golden Wonder Kilis. And all I can tell you about them is they're very excited to die because they jump like no other fish. They really want to be expensive dog treats. Geek Boy says, no evil plant murders. I don't know what we were talking about, but save the plants, man. Johnny says, you should rename the stream to Sex Talk with Kelly. I mean, I do talk a lot about sex because it really is, someone once told me that, like, you can divide all of biology into either eating or fucking. I mean, it's one of the 2 things. So, a lot of times, biology is a 2-semester sequence in college. You take biology 1 and biology 2, and you never really know how to divide that up, like what topics to put in each one. And one biology program had, like, the first semester was the flow of nutrients, the flow of energy. So you'd have things like respiration, photosynthesis, digestion, growth. And then the other one was flow of information. So you would have evolution, genetics, immunology, things like that. So everything's either eating or fucking. That's pretty much all biology is. All right. Sarah says her guppies they're in heat and ugly if in dark tubs. I have never had a guppy. Fun fact about Kelly, I have never had a guppy, even as a little kid. For some reason, my dad is very anti-guppy, and I don't really know why that is, but it's kind of rubbed off on me. I too am anti-guppy. Steven keeps threatening me with guppies, though. Brother Dan is actually retired, like all awesome people are. Suddenly I'm not awesome. I have a job. Yeah. Oh, Sarah's son turned 25 last night. You guys had mahi mahi. That is quite the treat in Iowa because Iowa is very landlocked and mahi mahi is expensive, but it's very good. You also get cheaper auto insurance when you're 25. That's true. So Jenna says I very little Gulf seafood when I was in Louisiana. Did I? I think I only ate seafood. I had like oysters and shrimp and I had snapper and I love seafood. It's so good. It's so good. It's delicious. So Dennis says Darwin's finches would be fast evolution. Yes, yes. They so for those of you who don't know, the finches in the Galapagos it's hard to say. The Galapagos Islands have all had a big adaptive radiation in a very short amount of time. And when Darwin was researching the natural history of that place, he took note of all of the variation of the finches and how they had evolved, which, he didn't use the word evolved at the time because he didn't know about that yet, but how there were so many changes to fill different dietary niches. So, for instance, you would see like a finch with a big beak would be for crushing big seeds. And then you'd have ones for like little bit like long pointy beaks for getting insects. And so there's just a huge number of these finches, and it's a very sort of classical example of adaptive radiation. And that happened in a pretty short amount of time. I don't think a short amount of time though as African cichlids. I can't think of any example of, at least in animals, we're not counting like bacteria or anything, but like in animals of any group evolving so quickly. Steven says blame the vegetables. I mean, maybe. Jake and I did eat the same vegetables and fruits though. So, man, I was so sick. It sucked. It sucked. Fish Dreams has only 1 month left in your 40s. Hey, you'll be nifty 50s. Daniel says, didn't have to know your real age. I mean, you shouldn't be ashamed of being 25. You have a lot of learning and growing to do. Geek Boys says, send all the glowfish you find to Kelly. The pictures. The pictures. Do you not want your actual glowfish? Fish Dream says Western medicine is lagging behind big time. Rant over. I mean, we're doing the best we can, man. We're doing the best we can. Core Work says carcinization is the recurrence of the crab body shape in evolutionary time. Oh, I think I have heard of that or something like that. It's like the joke that everything evolves into a crab eventually. I have heard that before. And then there's something about how they thought aliens would be crab shaped because it is such a common body plan. And if you think about it, a crab is very good at filling a lot of habitats. There were a lot of really cool crabs down in Costa Rica that I saw. I took some cool videos of crabs. I'll show them one of these days. So Johnny says 23andMe got my Myers-Briggs personality test right. How do you explain that? Sheer coincidence, because there is nothing more pseudoscience-y than Myers-Briggs. It's like astrology for office workers. I mean, you could take that test on different days and get completely different answers. That's what I would say. There is really no scientific validity to Myers-Briggs. But in case you were wondering, I'm an ENFP. So Corework says, did you verify it with your phrenologist and horoscope first? Yeah, that's about the truth. Yeah, that's yeah. Sarah says, my family found a 40-year-old unknown offspring. Someone traveled the country in a rock and roll band. Oh my. And I have heard of stuff like this. So my friend Mitch, he found a half brother in Indiana. His mom his mom and dad, they gave up a child for adoption before they got married. They were like 17, and they just never told him. And I guess, I mean, he and his long lost brother have gotten reacquainted, and they have become very good friends, and the families are all brought together now. And it's, I guess, a beautiful thing. But I guess for every one beautiful thing, there's some stories about mom cheating. I mean, and dad sleeping around. Scuba Sivo is doing his water changes. You got to do it. You got to change that water. It's important. Mickham says, if I'm correctly informed, DNA info on 2 separate species based on a new thesis for opistos. One was found in southern Peru in pretty high altitude, and the other in northern Colombia, Venezuela, also at high altitude. That's pretty interesting. Yeah, I mean, I don't know a whole lot about opistos either, but if they're in different habitats, I guess it would make sense that they are separate species. I mean, those waterways probably don't have a whole lot in common right now. If they're in different countries, they probably don't connect really easily. So, yeah. Matt says, don't knock online DNA. Would never have found my long lost sister mom. You know, it's bringing families together and tearing families apart, but I guess it's making it interesting. So in all seriousness, we did found out Stewie's 100% Alsatian wolf dog. I mean, that is kind of cool. Like, you know, the dog is some kind of mutt. We think she's mostly miniature schnauzer, but we don't know. I mean, my friends have gotten their dog breeds done. It's kind of fun. I mean, you know, what does it cost to get it done? It's not that much money. I've wasted money on way worse things. You know, I have bought some dumb shit in my lifetime. So if it makes you happy, go for it. Procreation with Kelly. I have never procreated. I would say that I know of, but like I would remember. So Matt says, we didn't do it for the breed. We did them both because Stewie has a major issue that has already cost us a ton to diagnose and treat. Yeah, and it's an amazing thing to have that information. If you have health problems, there may be very good reason for you to get your DNA sequence. You know, like I talked about, cancers running in your family. That's true for pets too, and it's, you know, it's a really amazing thing. If I had a reason to sequence my own DNA, I would. But I don't think I do. So Mickham says both high altitude opistos are closer related to each other than to any other closer living opisto. Land rising separated them apart for thousands of years. Yeah, I mean, you know, at one point they must have been together if they're closely related. But now they're not. The geography has changed in the intervening time. So kind of interesting. So too bad what he has wasn't found in DNA. That's unfortunate. Poor Stewie. He's a good boy. So Fish Dream's daughter just turned 27 and we had salmon. Oh, happy birthday. That's a good year. How many mahi-mahi can fit in a 10-gallon tank? They're big fish. I mean, so when we were down there before I got sick, one of the cool things we did is we went on a kayak tour of a mangrove swamp, which was really, really cool because the one we went to was just really pristine and beautiful. Like there's no trash anywhere in it. It was really just a cool habitat. And there's so many animals in the mangrove swamp. But there was a tour coming back of sport fishing, and they had a mahi-mahi they had caught that was just, it was like, I don't know, 3 feet long. Just a huge fish. So I think you could fit like 10 or 12 of those in a 10-gallon. Alex says, do platies instead of guppies. I've never had a platy either. When I was a kid, I think we did have some black mollies at one point. I don't recall them breeding. They died pretty rapidly. But I haven't had any live bears on my own. I don't think I will. I don't know. Maybe I'll change my mind though. You never know. So, what about the tilapia in the benthic lakes of California? I don't know anything specific about those, but I don't think that tilapia are from the US, right? Aren't they African fish? So the tilapia were probably released there, I'm guessing. I don't know the story about this. You're gonna have to help me out with more information because I don't know anything about those. I'll take the actual glowfish. I dig them. I will send them right to you, and you will send me back the pictures, and that is how that will work. So they are banned in the EU. That's interesting. So the EU has much more stringent laws on transgenic organisms, um, for good or for bad. I mean, I'm pro genetically modified organism uh, as a principle. I think they're much better for the environment than the alternatives. I eat GMOs with abandon. It doesn't bother me, but the EU has pretty strict laws about that. Some things the EU is more strict on and some less. The FDA for drugs is much, much more stringent than the EU. But for devices, medical devices, the EU is 1,000 times more strict than the US. So it's just different regions have different laws. Lady Rorschach is at the party. Oh, sick dogs and a mentally unstable person. Oh, I'm so sorry. I hope your dogs feel better soon. I hope the mentally unstable person causes you less grief soon. I'm very sorry to hear that. Okay, Alex says 23andMe and YouTube are both headed by nepotistic relatives of the founders of Google. My sister got it done and it was pretty worthless. Yeah, I mean, there are some things that are valuable to get done. Um, if you have a family history of certain genetic diseases, I think it is probably valuable. But otherwise, I don't know. That information is mine. If they want it from me, they should pay for it. I'm already giving enough of my information to Google for free. They don't need my genotype. So Coral Work says my great grandma never married, but she did have a number of trucker boyfriends she would go disco dancing with. At least so the stories go. She sounds like a fun lady. I love it. Dee Dee says, come to find out, I'm not who I always thought I was. Well then. I mean, Dee Dee had her scooter Putin phase, so we've all had our wild years. So her kid, my mom's dad, did 23andMe and went on a tour of the US to find all his cousins and stuff because he never knew his dad. He made like a scrapbook memoir about it with the family tree. I mean, it can give people an instant family. I mean, it can be good. Yeah, she sounds like she seemed to have a good time. Those disco years were wild. So Dee Dee says the one I always thought was my grandfather was a terrible person. Well, that's good because you're a pretty good person, Dee Dee. So, oh, yeah, I'm sorry I'm sorry going through some troubled times. I hope your dog gets better, Lady R. Bunny Viper says, just send those glowfish to me and you'll take the pictures. Yes, I don't think I want to keep glowfish for myself. Just want to talk about them. So Corework says, I remember reading about reservoirs in California that used to be freshwater and have become more and more saline over the years, which have tilapia that have adapted to the saltier than the ocean lakes. That's very interesting. I don't know if they have genetic changes. I'm not sure. I mean, fish can adapt to, fish can change their physiology somewhat to adapt to different salinities. It's not a genetic adaptation, it would just be a developmental change. But maybe they have undergone some selection in a new habitat. I don't know. Um, I don't know that they would be a different species yet. One of these days, we'll have an episode of definition of species, because that's something that is always interesting to talk about. So Corwork says, and I was curious if that was epigenetic adaptation or whether it was evolution. So I don't know. So an epigenetic modification is not a change to your DNA sequence. It is a change to the expression of your DNA. So those are epigenetic modifications. And maybe we'll do a whole stream on epigenetic modifications, because it is something I'm very interested in. So Silver Creek says kingdom, phylum, class, order, family, genus, species. Yes, King Philip came over from Germany sailing. That's how I would remember it also. Yeah. What do you mean you don't know? But you were the dispenser of knowledge. I don't know everything. There's just so much to know. My area of expertise is really just molecular genetics. It's not animal evolution. I mean, there are a lot of people in the fish fam who know more about fish than I do, for sure, because I don't breed them, for one. So Coral Works also wants to talk about epigenetic stream. Yeah, I think that would be a pretty cool topic. And I am super passionate about epigenetic modifications. I mean, I did a lot of PhD work on that. I'm very, very interested in general about control of transcription. Just, yeah. Well, guys, I reached the bottom of the chat. So, I don't usually get to the bottom of the chat. I mean, I would say I'm disappointed, but you guys have asked me some pretty good things. So, I mean, I don't know what I can tell you now that I'm at the bottom of the chat. So some topics I've thought about. Maybe I'll run some topics by you of things that I've thought about talking about. So Jenna wanted to talk about neurotransmitters. So I thought maybe we would do that one of these days. But let's see. What are some other topics I maybe had? Parasites and autoimmune disease. I thought that would be interesting. There's actually a real correlation between the disappearance of like gut parasites, so worms, and the emergence of autoimmune diseases like IBS and Crohn's disease. And there's this guy at the University of Iowa who did a lot of and he's at Harvard now because, Harvard scooped him up, treating diseases like IBS, which are very resistant to treatment. If any of you have IBS, it's really difficult to treat, but they're treating it with an intestinal parasite called pig whipworm, which is like a teeny tiny little 1 centimeter worm that they infect the person with. And then on average, they have 7 or 8 of these little wormy worms living in their body, and their symptoms pretty much disappear. And yes, IBS is an autoimmune disease. It's a disease of the mucosal immune system. So, yeah, and there's all kinds of autoimmune diseases that really were not a thing 100 years ago. And a lot of that is because our immune systems don't have anything better to do than attack ourselves. It used to be that keeping the parasite load in check was a big part of your immune response, because you would counter parasites all the time. Everybody would have everybody would have a parasite or two in their body and be fighting it off. Well, now that we don't have that, your autoimmune diseases, boards. You get things like allergies and asthma and rheumatoid arthritis and IBS. And these are kind of diseases that we have in environments with good sanitation, hypersanitation, and that they don't have in countries that don't have good sanitation. I mean, I don't want to give up good sanitation. I really don't want to have a nasty worm parasite because those kill you. But like all things, there's trade-offs in life. So, yeah. So that's one thing I thought about talking about. Other topics. Let's see. I thought I could talk about hair. Hair is interesting. We could talk about like the biochemistry of hair. We talk about like how different shampoos work, and like there's this stuff called Olaplex. We could talk about how that works. Let's see. I really want to do a couple, maybe one, maybe even two streams on photosynthesis. I think Geek Boy asked for that because for those of us who like to grow plants, knowing more about photosynthesis is pretty exciting. Other topics, fabulous fermented foods. I mean, fermented foods are pretty exciting. Talk about that. CRISPR Cas9. Maybe you've heard about it before. You've heard it on the news. So CRISPR Cas9 is a really awesome way to edit genes. And it is like a 10-year-old technology. I taught a whole course on CRISPR Cas9. And it's really just changed so many things in medicine. So I figured we could maybe talk about what that is. And then, maybe, I don't have a lot of topics, but if you have any topics, let me know. So, yeah, let's see what the chat has talked about. So Fish Dreams, wants to talk about hormones as a topic. Yeah, I mean, so neurotransmitters, they're pretty similar to hormones, but yeah, we could talk about them. So hormones, by definition, what they are small secreted molecules that have effects throughout the body. So not just testosterone and estrogen, but there are other hormones too. There's also plant hormones. We could talk about those. So Fish Dreams would like to talk about viruses. We could maybe talk about viruses. Viruses are pretty cool. Oddly enough, I know way, way more by like a factor of 10 about viruses that attack bacteria. So they're called phages than I know about other viruses. So yeah, and that's because I taught genetics for so many years. Matt would like to know where babies come from. Well, we don't need a stream for that because I can tell you it's the stork, Matt. The stork brings the baby. So IBS is an autoimmune disease. Yeah, it's an autoimmune disease. Yeah. So Corrick says, I have undiagnosed tummy troubles and I'm afraid to find out it's Crohn's disease or something that can't be treated for $300 a month. Yeah, I mean, all those gut maladies are really, really hard to treat. There's some really good success with fecal transplants. Gross as it sounds, you could have a microbiome that doesn't have the right bacteria in it anymore for whatever reason. And some of those, there's been some success with treating patients that way, but they are really hard to treat. Celiac disease, that's another autoimmune disease. So much more celiac than there used to be. So celiac is where you can't eat gluten. 100 years ago, celiac was very uncommon. Even when I was a little kid, I'm not that old, celiac was really uncommon. And now I know several people who have celiac and they are just straight up hospitalized if they have any gluten. It's pretty scary. So Nona does not have IBS, but other autoimmune diseases, those are just so difficult to treat, so difficult to diagnose. They're really a challenge for both patients and physicians. They're just, yeah, they're really, really difficult. So Silver Creek says phylogenetic balance pertaining to subterranean species and humans. I don't know exactly what you're trying to ask with that. I will say that humans don't really have any subterranean evolutionary modifications because we have never had any populations that have lived underground. But there's some cool examples of organisms that have lived in caves, like of the Mexican cave fish tetra. Those are pretty cool. There's other things that live in caves. Troglodytes, that's what they're called. Cave dwellers are troglodytes. Corework says incurable but treatable is my biggest fear. Now it's a pre-existing condition and you can't change insurance policies and all that horror. Well, under the Affordable Care Act, so also known as Obamacare, they cannot ask you pre-existing conditions when you apply for insurance. You cannot be denied coverage because of a pre-existing condition. So at least you can put that fear to rest. So that's good. But yeah, having a chronic condition sucks. I mean, but people are working really hard on those, and who knows what treatments there will be in the next 10 years. I mean, things like microbiome therapies and fecal transplants sound gross, but they're changing lives. Things like parasite therapies, there's all kinds of things happening. So I think there's reason to hope. Matt has been told that he is a parasite. Is that the same? I mean, you are a parasite. It's true. I mean, you're a beloved parasite, but a parasite nonetheless. Skinny notices colostomy bags aren't sexy. Go to the doctor. Amen, sister. And I would tell everyone who is in this chat, go to the goddamn doctor. Go every year. Get your physical done. Take care of your health, especially all the men in the chat. Do not put off health decisions. Take care of your bodies. You only have one. Rant over. So Corework says, my buddy was being treated for IBS for months before they realized it was just his appendix acting up. Difficult to diagnose, difficult to treat. I'll say that about it. Yeah. Those appendices are, yeah. Bunny Viper out of nowhere says, did you know that ducks have a 9-inch corkscrew penis and the female's vagina corkscrews in the opposite direction? Just learned it the other day. Holy smokes. Think about condom technology for ducks. How difficult is it to put on that condom? Corrick says, I don't know. I trust medicine as a science. I don't trust doctors as individuals or insurance or whatever as industries. The insurance industry is straight up sucks. I would encourage you to find a doctor that you can work with. Maybe a nurse practitioner. Maybe that's a good route for you. But find one that's good. Don't go to a doctor you don't trust. Find one you can work with. I go to a nurse practitioner and I love her. She is great. Dee Dee says, I had ducks before and I learned it then. First I was like, what the hell was that? God, that's got to be something, Dee Dee. Like, what is wrong with my duck? Geek Boy wants his own CRISPR. I mean, it is pretty cool. Bunny says, CRISPR is awesome. Love to hear about it from you. Yeah, I mean, we can also watch like videos about how it works and I can narrate it for you. It is very cool technology. And the cool thing about it is before there was CRISPR, site-directed mutagenesis in most eukaryotes was like impossible. So the site-directed means like you decide where the mutation goes or what piece of DNA you're cutting. That was pretty much impossible. But you know what organism you can do it in is yeast. Yeast has had site-directed it just naturally has it. It's a gift of the gods, and that's partly why I worked on yeast. As genetics is so incredibly easy in it. But now it's easy in other organisms too, and it's really changed things. So Sarah's, I didn't know your appendix could act up. I thought it was either okay or really bad. Well, I guess like there's like stages on the way to imploding it goes through. I mean, yeah, I mean, I would assume if it's starting to act up though, they just take it out because they don't want to mess around with that. I mean, if it goes necrotic, it can kill you. So, I think that if it's starting to act up, out it goes. They don't mess around. Dee says the guy was showing it on a video when he squeezed the duck. What are you looking at this duck pornography for, Bunny? Let them do that in private. Dee says Drake has a lot of work cut out for him. Just think if a predator approaches when his corkscrews out. Difficult times. Corework says he doesn't have his appendix anymore, so I'm assuming it was bad. Yeah, they don't mess around with that. I mean, they would just go ahead and take it out because the risks are pretty significant. So Tommy says, I have a cousin that just died from an unknown autoimmune disease at 42. She was the lifeblood of our family. I'm so sorry to hear that. And yeah, autoimmune disease is difficult to diagnose, difficult to treat. And they're something that, are kind of on the rise. There are so many more of them than there used to be. They were not an issue 100 years ago. So Coro says, I've tried cutting a whole bunch of things out of my diet in stages, but it's been years since I really put that much time and effort into managing it. Difficult to diagnose. Difficult to diagnose. But there are some tests that they can, just like allergen tests. And there are other autoimmune disease tests that they can give you to help diagnose things. So, go to the doctor. You should go. I'm getting at aliens. Well then. Sarah says my hair is falling out from my resent plate. Oh dear. So Jennifer says, pheromones to initiate spawning. Eric Bodrock has a system that trickles water from spawning corridors to non-spawning bears. He's a clever, clever man. What an incredible fish breeder. He's a great person to get rainbow fish from, by the way. He is he All Oddball Aquatics, I believe, on Aquabid? Yeah, All Oddball Aquatics. Terrific fish breeder. And his fish are really good quality. His good customer service. I bought rainbows from him and they were great. He's an amazing breeder. A lot of those things, we don't even know what the pheromones are. They hypothesize there are pheromones. There probably are pheromones. I have no doubt they're pheromones, but we don't know the exact biochemical structure because a biochemist hasn't fractionated it out yet. There's just so many things we don't know in the natural world that are to be learned. Maybe we don't know everything there is to know about Corydoras. So yeah, but he is such a cool breeder. Just a cool guy in general. More about ducks. Cool. Alex says 2 women came up with CRISPR, one at Cal and one French person. So yeah, Jennifer Dewadna and something La Charpentier. Yeah. And then a Chinese idiot tried to steal the country. No, because he actually worked on it independently too, and I believe they have all shared the Nobel Prize. What was his name? Well, if I do a stream on it, I will tell you all their names. But I have seen a talk from Jennifer Dowadna, and she's a really smart lady. She's a really smart lady. Yeah, Tommy, I'm so sorry. That's so hard to lose someone you love. Troglodytes are freaking cool. It is true. They're really cool animals. So Tommy also has another cousin with lupus, and I can't remember, but she can't eat meat protein, and now her bones are super weak and break easily. Yeah, difficult to treat. Difficult to treat. Sometimes diets work, sometimes they don't. Um, and maybe it's lupus, maybe it's another autoimmune disease that presents like lupus. Those are very challenging. Those are very challenging conditions for both the patients and the doctors. Corwork suggests we transcend the flesh and move into robot bodies for the metaverse. I really don't want to move into anything owned by Facebook because they are just the dumbest company. I mean, every change they make to Facebook makes it worse. I don't want to trust them with my body, with my consciousness. Dee Dee loves hearing me use them $3 words. So Jennifer worked at Penn Vet School in large animal reproduction. You had the best Halloween parties. I bet you did. I bet you did. I could never be a vet tech. I am too tenderhearted. I would burst into tears all the time. Bunny Viper says all these mental and physical ailments that seem to continue exponentially. What do you think causes them? What about preservatives in our food? Any thoughts? Well, I mean, preservatives are a double-edged sword, right? I mean, eating food that is riddled by bacteria and fungi can make you really, really sick. But maybe preservatives aren't good either. It's a double-edged sword. I would say in general, most Americans eat pretty crappy food. Not always sometimes that's not their fault. If you don't know any better, if you don't have time to cook, if you don't have access to a grocery store, if you don't know how to make things, can't really fault you for it. Also, shitty food tastes good. Turns out, it just does. Puffy Cheetos are terrible food, but they taste good. So yeah, I don't know that physical ailments are increasing exponentially. I mean, our lifespan, well, for the longest time, it was getting longer, the average lifespan, but it has started going down. But that's mostly because of both COVID and the opioid epidemic. So yeah, I don't know. In general, compared to 100 years ago, people live longer lives. Women don't die in childbirth. That's pretty great. Infant mortality is down compared to 100 years ago. I mean, I'll take it. So, yeah. Mental ailments are a different thing. I think a lot of that is due to loneliness. A lot of it. People don't have close-knit communities. A lot of people are really lonely. So, forming pairs through pheromone therapy, essentially, Jennifer suggests. I mean, Eric Bodrock is a genius. He is, yeah. So I think it was Dean that uses a tank divider with breeding angels, and the pheromones inspire the upper couple. I would not be surprised. Dean Tweedale is also a genius fish breeder. I mean, he really does breed a lot of fish. I breed almost no fish. I am not an expert at fish breeding. The only fish I have ever bred are my Medaka rice fish. I did nothing. They just bred. But Jennifer Weaver is a pretty talented breeder, so she's playing around with it. She knows more about fish breeding than I do. So Corework says even ignoring the preservatives, the processed nature of a lot of shelf-stable and ready-to-eat foods contribute to gut and dental issues, and that's documented. Yeah, for sure. I mean, processed foods are not really a new thing. I mean, people used to eat so much salt in their diets because of salted foods. Smoked foods have carcinogens in them. Grilled foods have carcinogens in them. So those are not new things. Um, yeah, I mean, in general, people should eat more fruits and vegetables. They should eat more whole grains. They should eat less meat than they do. I mean, I eat meat too. I'm not like preaching you have to be vegan or anything, but we probably shouldn't eat as much as we do. You know, it's hard. It's hard to eat good food all the time. We gotta try though. So Sarah says I use Adaptil to calm my dog. I call it calm your ass down spray. It's synthetic pheromones that mimic the mother when nursing. That's very interesting. So I actually got some cat pheromones. Um, so you get this like it's like a little plug that you plug into your, an outlet, and it's supposed to calm the cats down because Big Kitty hates Tiny Kitty. And so he's been peeing in the corner in my bedroom all the fucking time. And like I have to put pee pads down there and he won't stop. And so I have tried these like I think it's called like Feel Away, which is supposed to be cat hormones. Anyway, I don't think it's done anything. Big Kitty just hates Tiny Kitty. Just really hates Tiny Kitty. Yeah. Cats, man. Cats. Dee Dee says, one thing that people have nowadays is a video of the loved ones. Looking at pics of my mama was one thing, but when I heard her voice, oh, yeah, that's true. I mean, that is kind of, kind of a thing that we'll have to remember people by. Silver Creek says Facebook is the devil. It's okay, you can say it. I just think they're also a really badly run company. They're a platform that tries to be every platform. Like they try to be TikTok and they try to be Discord and they try to be this and they do it all badly. And it's this weird like chimeric beast of a platform that kind of does nothing well. And I also think they're evil. I don't know that they're necessarily good for society as a whole. Oh, can't see your mom's handwriting without bawling. Yeah. Yeah. That's hard. Tommy says we eat a massive sugar and unhealthy fats, but damn it, Big Food won the day with that one. Love me some sweets. Yeah, I know. It's I try really hard to eat a lot of fruits and vegetables, and I like fruits and vegetables, and I also cook my food. But damn it, I ate puffy Cheetos yesterday, which objectively speaking is the worst food on the planet. There is no food more devoid of nutrients than a puffy Cheeto. But like two or three times a year, just gotta have them, right? Oh my gosh, guys, it's 9:34. Whew, we gotta go see Chattanooga Head. Everybody go see Chattanooga Ed. Hashtag Fat Kitty. Thank you to my lovely mods. Thank you everyone for being in chat. Thank you for your questions. Thank you for your comments. It's good to be back. Thank you to my lovely replay crew. Please leave a comment. I always answer your comments. And I'll see y'all next week. Bye.